Regulation of gene expression. In the previous lecture, I discussed how the cell builds new cellular components required to prepare for cell division and to maintain itself. The processes of DNA replication and protein production must be regulated so that the cell makes those components only when needed. In this lecture, you'll learn about how gene expression is regulated. You'll learn how the transcription of genes is turned on and off using two well-studied examples, the lac operon and the arginine operon. Then, I'll briefly discuss how mutations can be made in the DNA that may result in a change in the protein. Genes aren't transcribed and translated until the cell needs a particular protein. So there has to be something regulating the start and stop of transcription. In prokaryotes, genes with a common function are grouped together in areas on the chromosome called operons. The operons are controlled by regulatory proteins called repressors, and the genes that code for them are usually near the operon. Pictured at the top is a generic operon. You can see there are four genes, genes A, B, C, and D. In green, these code for proteins A, B, C, and D. When operons are transcribed, one strand of mRNA is made for all of the genes in the operon. Along the mRNA are start and stop codons for each protein, so the ribosome knows where to start and stop translating each protein. In front of the genes is the promoter sequence, where the RNA polymerase binds, as seen in the previous lecture. Here, there is an additional site on the DNA called the operator. This is where regulatory proteins called repressors bind to block transcription when the proteins are not needed by the cell. Repressors are like huge trees blocking a road. When the tree is there, the car can't drive through. But once it's removed, the car is free to drive down the road. The promoter, operator, and structural gene sequences are all part of the same operon. There are two types of operons to know: inducible operons and repressible operons. Inducible operons are off until they are turned on by the substrate. These types of operons are catabolic, meaning the proteins they code for are used to break stuff down in degradation reactions. For example, enzymes that are needed to metabolize a nutrient are only produced when the nutrient is present in the cell. An example of an inducible operon is the lac operon. Repressible operons are always on until they are turned off by the product that is synthesized. These types of operons are anabolic, meaning the proteins they code for are used to produce proteins that are always needed by the cell in synthesis reactions. For example, cells are constantly making proteins and always need amino acids. An example that we'll discuss is the arginine operon. The genes in the lac operon are not transcribed until lactose sugar is transported into the cell. Otherwise, transcription is off. So the lac operon is an inducible operon. It is always off until it's turned on by lactose, which is the substrate. The top figure shows the lac operon. It consists of three genes, LAC-Z, LAC-Y, and LAC-A. The proteins that these genes code for are used by the cell to break down lactose. So when there is no lactose in the cell, the genes don't need to be transcribed and translated into proteins. The repressor sits on the operator to block the transcription by RNA polymerase. So RNA polymerase can still sit on the promoter, but it can't transcribe the genes because the repressor is blocking it, just like the tree blocking the road. When the lactose substrate enters the cell, it binds to the repressor protein and it changes shape so that it's no longer able to bind to the operator and falls off of the DNA, just like when removing the tree from the road. Now the RNA polymerase is free to transcribe the genes needed to break down the lactose. When the cell uses up the lactose, the repressor goes back to its original shape and binds the operator again to block transcription. You should understand this process and be able to problem solve different scenarios. For example, if the repressor is mutated and can't bind the operator, or if the DNA sequence is mutated where the RNA polymerase can't bind to it. The arginine operon is a repressible operon, the opposite of the inducible operon. The cell is constantly making the amino acid arginine because it is constantly making proteins that require that amino acid. So the arginine operon is always on until it's turned off by the product. This figure shows the repressor, circled in red, off of the operator, and RNA polymerase is free to transcribe the genes that are needed to make arginine. When the levels of the arginine get too high, the arginine product binds to the repressor and it changes shape so it can bind the repressor and turn off transcription until the cell uses the arginine and the levels drop again. So again, the arginine operon is always on until turned off by the product. The arginine is considered a co-repressor because it helps to activate the repressor. You should also be able to problem solve any issues that may occur with this operon if there are mutations that prevent its normal function. Speaking of mutations, they can occur when there is a change in the DNA sequence, meaning a change in the genotype. This usually leads to a change in phenotype, which we can refer to like the cell or colony morphology, metabolism of the organism, or resistance to chemicals like antibiotics. You will learn about point mutations and how the change of a single nucleotide can cause major problems. In genetic research, the wild-type strain is the strain of bacteria that is designated as normal versus the mutant that has some type of problem and makes it unable to perform normally. You've learned that the ribosome reads the messenger RNA 3 bases at a time to determine what amino acid to incorporate into the protein. Most mutations happen at the DNA level, where there are bases that have been deleted or inserted, and these additions or insertions throw off the reading frame for the ribosome. A point mutation is when a single base is affected. A base is changed, inserted, or deleted from the DNA sequence. Pictured here is an example of a missense mutation. This type of point mutation is due to a change in a single amino acid. One base has been changed, and the new sequence codes for a different amino acid. The sequence on the top in the red square is TGG and codes for the amino acid tryptophan. Something happened where the guanine was changed to a thymine in the DNA. So now, when this new DNA sequence is transcribed and then translated, the new amino acid is cysteine. Recall that proteins need to be folded properly in order to function properly. The change from tryptophan to cysteine may not allow the protein to fold in its normal shape, and therefore the protein would not be functional normally. An example of a point mutation that occurs in humans and causes major problems is sickle cell disease. There is a single base change in the gene that codes for hemoglobin, and the hemoglobin can no longer normally carry oxygen and keep the cells in their normal spherical shape. Nonsense point mutation changes a normal codon to a stop codon. Here, the guanine is changed to an adenine, and when transcribed and translated, results in a stop codon, so the protein cannot be fully translated. Silent mutations are the most harmless. Recall the genetic code is redundant, and most of the amino acids have more than one codon. Silent mutations change a single base, but due to the redundancy, the amino acid remains the same. In this example, the thymine was changed to a cytosine, but the amino acid is still tyrosine. Frameshift mutations are those that add a single base or delete a single base from the DNA. These mutations throw off the reading frame the ribosome follows, changing all of the codon from the point where it appears in the sequence as well as afterwards. In this example, there has been an insertion of a cytosine. The ribosome still reads the bases 3 at a time, but note how all of the amino acids have changed. Mutations can be spontaneous. Random changes in the DNA happen all the time during replication, but when they aren't corrected, they lead to problems. Sometimes in research, we want to create mutations so we can study the function of different genes. This table shows a number of ways researchers make mutations by using different forms of radiation as well as chemicals. Mutations can be harmful or fatal to the cell if the protein the DNA codes for is essential. Other mutations can be beneficial if they allow organisms to adapt to a changing environment or to survive exposure to certain chemicals like antibiotics. You should understand the different types of point mutations and the connection of DNA to the protein. This is the end of the lecture. Thank you for listening.